Hello, respected ladies and gentlemen. I am Ronnie Chandamandal, welcoming all of you to my video with an interesting topic, which is prebiotics in aquafeeds. Simply, prebiotics are good bacteria promoters and served as food for beneficial bacteria or probiotics, which eventually provide health benefits to the hosts. Considering the negative impacts raised by prophylactic and therapeutic use of antibiotic in aquaculture, administration of functional dietary supplements like prebiotic received an increasing attention as an environment-friendly strategy for improving fish health. There are some interesting functions of prebiotics related to growth and disease resistance, and therefore we need to take account of in nutrition. Let's have a look in detail. In this video, I am going to introduce some commonly used prebiotics in aquafeeds and their importance related to nutrition. And also, I am going to explain the mechanism of function of prebiotics in gut and how they play a role in improving health. Here's the outline of my presentation, and I'm going to talk about these topics in the rest of my video. A prebiotic is mainly complex carbohydrate, act as a functional dietary supplement, which is soluble and non-digestible fiber that beneficially affects the host by selectively stimulating the growth and the activity of one or a limited number of bacteria, particularly lactobacillus and bifidobacterium species, and also by reducing of bad bacteria, potential pathogenic bacteria such as Aeromonas or Vibrio species in the gut and thus attempt to improve host health. But why is it important in aquafeeds? Well, prebiotics plays a number of interesting functions which are outlined in the right side of the slide, but most notable functions are it stimulates the growth and reproduction of beneficial gut microflora, suppresses the reproduction of pathogenic organisms in intestine, maintains optimal pH, removes excess mucus from intestinal cell wall, and stimulates local immunity and disease resistance. Therefore, putting prebiotics in aquafeeds eventually improves growth performance, survival, FCR, digestibility, gastrointestinal enzyme activities, and immune functions of finfish and shellfish. Then the question is, which complex carbohydrates are commonly used in aquafeeds? Here is the list of commonly used prebiotics in aquafeeds. Mainly, prebiotics are classified into two major groups, that is, oligosaccharides and polysaccharides. In oligosaccharide groups, mannano-oligosaccharides and fructooligosaccharides are the most studied prebiotics, whereas inulin and beta-glucan are the representative of polysaccharides. Most of the prebiotics are plant-derived, whereas some of them are derived from yeast cell wall. Other than these, there are some commercially available prebiotic products which are made in combination of yeast byproducts with different oligosaccharides and polysaccharide-based prebiotics. Let's see the mechanism of action of prebiotics in the gut. Once prebiotics enter into the gut, they are selectively metabolized by the gut commensal microbiota. This step increases bacterial growth and functionality of specific genera or species. This gut microbiota can ferment prebiotics into short-chain fatty acids called metabolic products including acetic acid and lactic acid. These short-chain fatty acids lower the luminal pH, provide energy sources for epithelial cells, and have profound effects on inflammation modulators and metabolic regulations. These metabolic products can also influence epithelial integrity and hormonal regulation. A well-balanced bacterial community can also improve intestinal mucosal structure. Some bacterial strains produce antimicrobial factors or stimulate the immune system by signaling dendritic cells. The control and integration of this immune response is carried out by cytokines, which are mainly produced by lymphocytes and macrophages after stimulation. Prebiotics can reduce pathogen colonization by blocking the receptor sites used by pathogens for attachment to the epithelial cell surface. All their beneficial effects are the result of a complex set of interactions on host physiology which result in improved productive parameters of host. Let's have a look in details on some important prebiotics. The first one is mannano-oligosaccharides, which are indigestible and short-chain oligosaccharides. The addition of mannano-oligosaccharides in diets stimulates the immune system by increasing the activity of immunoglobulin M, which is the first measure of antibody responses to disease that animals tend to have in their body, and also by increasing the lysozyme activity, which is an antibacterial enzyme that is generated by animals to combat disease. Mannano-oligosaccharides provide mannose-substrate upon which pathogenic gut bacteria selectively attach. The inhibition of pathogenic bacteria adhesion to enterocyte prevents the formation of colonies, leading to better gut health by increasing regularity, height, and integrity of gut villi, and consequent better utilization and absorption of nutrients. Some studies have also shown that adding mannano-oligosaccharides in diets provides more mucus coat in fish, which makes them more resistant to sea lice. Moreover, dietary inclusion of mannano-oligosaccharides has shown positive effects, including increasing lysozyme activity, disease resistance against Vibrio and other pathogenic bacterial species, and growth performance of rainbow trout, European sea bass, and tropical spiny lobster. The graphical representation shows the productivity outcomes where the average harvested biomass and yield of white-leg shrimp found higher in ponds supplemented with mannano-oligosaccharides and in ponds with commercial diet. The next one is fructooligosaccharides, which are short and medium chains of beta defructans in which fructosyl units are bound by beta-2,1 glycosidic linkages and attached to a terminal glucose unit. Dietary inclusion of fructooligosaccharides could positively shape the gut microbial community and then modulate the immune function of fish. The effect of fructooligosaccharides on the innate immune response is found significantly higher in some fishes like Caspian roach fry, red drum, and sturgeon juvenile, whereas no significant impact found on the lysozyme activity in Japanese flounder. These contradictory results may be attributable to different doses, different duration of fructooligosaccharides administration, different fish species, or life stage. The right graph shows that lysozyme activity of sturgeon juvenile differed with different levels of oligosaccharide. Inulin is a heterogeneous blend of fructan polysaccharides with the degree of polymerization greater than 10. Several studies have evaluated the positive effect of inulin supplemented diet on the reduction of micrococcus bacteria in Atlantic salmon, increase in lysozyme activity, immunoglobulin M response, and resistance against streptococcus species in rainbow trout, increase in growth performance and resistance against Aeromonas hydrophila in Nile tilapia, and increase in survival of Indian white-leg shrimp post-larvae. In fish, 1316 beta-glucan injection leads to an enhancement of the immune response such as macrophage activities, which stimulate the phagocytic cell of the innate immune system that are located in various tissues to engulf pathogens. When given orally, glucans are able to stimulate the immune response and disease resistance of fish. Beta-glucans promote intestinal health through increasing mucus secretion and can also be used as feed additive. The right graph shows that beta-glucans with high doses of vitamin C have the stimulatory effect on phagocytosis in rainbow trout for a short feeding period. Despite the potential benefits to health and performance as well known in various terrestrial animals, the use of prebiotics in the farming of fish and shellfish has been less investigated. Moreover, the efficiency depends on a number of independent and interacting factors, for example, age and size related responses, appropriate doses, and timing of administration, which are specific to the culture conditions and host species. Therefore, greater attention and future research to elucidate these causative actions are needed to improve the overall efficacy of prebiotic applications. That's all about my presentation, and thanks to all respected viewers for your patience watching.